Welcome to everyone. So I'm Hugh Patterton. I'm the Director of the Centre for Bioinformatics and Computational Biology at Stellenbosch University, and we're very pleased to welcome you on behalf of Stellenbosch University and more specifically the Faculty of Science and Science Café at Stellenbosch. Before introducing the speakers, I would like to share a few house rules. We're recording this talk. We will keep to 1 hour strictly. The speakers will address us for 30 minutes, and then we'll have time for some questions and discussion until 7 p.m. And please mute all your microphones and switch off your cameras during the entire presentation period. Please address your questions to the speaker in the chat section on Teams, and we will address as many of them as is possible. And you're very welcome to actually send the questions during the presentation. You don't have to wait until the presentation is done. The topic for this discussion tonight is developing a COVID-19 risk prediction platform using sewage water. So we are very fortunate this evening to have as our speakers 3 people who've been working at the coalface since the outbreak of the pandemic in South Africa early in 2020. They are Professor Wolfgang Preiser, who's from Medical Virology Division and at the Faculty of Medicine and Health Science, and is by now a household name in South Africa. And with him also from Stellenbosch University is Dr. Edward Archer. He's the coordinator of the South African Collaborative COVID-19 Environmental Surveillance System with the acronym SUCCESS. And he's also a postdoctoral fellow in the Department of Microbiology at Stellenbosch University. And then lastly, we have Professor Rabia Johnson. She's an associate professor at Stellenbosch University, and she's also a deputy director at the Medical Research Council, where she's working at the Biomedical Research and Innovation Platform. In this discussion, we are posing the question, what if we had an early warning system in place for all types of contagious diseases? Now, for alerting us to the onset of a possible third wave of COVID-19 infections, and in future, we may be faced with similar outbreaks. So our speakers will explain the involvement with efforts to establish a wastewater-based surveillance system for contagious diseases such as caused by the SARS-CoV-2 virus, the science behind the environmental fingerprinting of the disease, and whether the current system is ready to predict a third wave. So it's a pleasure to welcome the speakers and the audience, and I'm handing over to Professor Wolfgang Preiser, and we're looking forward to hear what you have to say. Wolfgang, please go ahead.
Thank you very much for the kind invitation and for the friendly introduction. It's a great pleasure doing this talk. I will start with the boring part, and that is to present where we are in terms of epidemiology and surveillance. So I will mention everything but the actual topic of tonight's Science Café. This is my division, the top floor here in Tygerberg on the Cape Flats, not in beautiful Stellenbosch, of course. And this slide shows the gold standard for diagnosing SARS-CoV-2 infection, i.e. COVID-19 patients. It's the rather unpleasant nasopharyngeal swab that gets inserted into the back through the nose into the back of the throat here, and then it's a rather difficult and cumbersome and demanding technical process that amplifies the viral genome and then detects it. It's a good technique, but as you could probably guess, it's quite demanding to do. It needs specific equipment and trained and qualified people. It's also expensive, and our capacity to perform this is very limited. And this is a picture of our reception area during the first wave of COVID-19 about a year ago. And you can imagine that if each of those contains about 40 to 60 swab samples, all of them need to be processed. And the moment that the lab has made inroads, this bench will fill up again, and here we start again. So it's basically very difficult to keep up. And there are some other limitations as well, and that is the clinical course of SARS-CoV-2 infection. People are infectious before the onset of symptoms, which is this line coming down here. And during that time, they are already infected. They can already pass on the infection, but we normally don't test them at that stage because they don't have anything to suggest that they might be ill. And as you will have heard, many people actually never ever develop symptoms that are associated with the infection. They may be asymptomatic, or it's so unspecific that they don't worry. So it is not that easy, and in addition to that, the labs during the peaks of the infection may just be overwhelmed. So how do we know how bad it is? How do we know how many cases we have? Well, there's a number of data sources. And I must say South Africa is actually doing very well in global comparison. The National Department of Health, you're probably familiar with the SA Coronavirus, COSA website, the National Institute for Communicable Diseases, that publish on a weekly or monthly basis a plethora of different surveillance reports using different data sources from hospitals, from surveillance cohorts, and looking at the tests being done by the National Health Laboratory Service, the SAMRC, the Medical Research Council, and of course I don't now want to jump forward, so I'm not mentioning the one dashboard that we are interested in tonight, but they also issue regular reports on the deaths occurring in South Africa. Very important if one looks at the fact that people may die from COVID without having been tested and therefore not being counted. And there is also the Academic Explorer Consortium, which is a modeling consortium where the Stellenbosch SACIMA plays a major role. So all of these are excellent sources of data, and this is the new type of release that one sees every night. The tests conducted, the positive cases found, that's cumulative figures up here, people recovered, which is the indirect, and the numbers of deaths and the numbers of new cases, which is really, what we want to look at. We want to look who is newly infected, what are the deaths that are occurring on the day. This is now from yesterday's figures, and now also how many vaccines have been administered. And there's a dashboard at the NICD, now coming closer to the Western Cape, and I'm fortunate to have quite good insights in sitting on all sorts of committees of the Department of Health here. This is one of the regular status updates, and you see, the numbers of different data that are available here, testing by specimen date, by reporting date. Remember, the PCR test takes time. It's quite demanding, so it's quite usual to have a turnaround time from taking the sample to getting a result of a day, very often longer than that. And during the peaks, when the labs are really busy, it may be 3 or 4 days, which then, of course, makes it quite unuseful, because by the time you get the result, the patient is almost recovered, hopefully. And then, of course, a breakdown according to area. And this is just a little glimpse of this. I can recommend having a look at the dashboard, which is also makes for interesting reading. Again, different functions that you can look at, cases over time, cumulative, the vaccines administered, a spatial distribution across the province, a list of active cases by sub-district. There's a lot of information out there. Now, if we take these different information sources together, as we are doing for the Western Cape province, you get something like this. And I just briefly want to go through this. It starts with the numbers of new diagnoses. These are the gray bars here, and you see very clearly the 2 waves. This is the first wave last year. This is the second wave at the end of 2020, early 2021, a lot worse. Importantly, the test positivity rate, which you also see here. So what proportion of tests being done was positive, which is meaningful in that you realize that, if that surges, you realize that there may be many more cases out there that are not getting tested if a large proportion of those patients that are sent for testing are positive. Then patients being admitted to hospital, patients needing oxygen, and patients dying. And you see that these curves sort of show slight delays from one another, and that's obvious because first you need to become infected. You have an incubation period. You become infectious, not yet ill. Then you become ill. The next day you go to a doctor. 2 days later you get a test result, and this is what the test reported day then means. A week further down the line, hopefully not, but in many cases you may be more seriously ill. You need hospital admission. You will, after a few days, be put onto oxygen, and if you know the events really take a tragic turn, then you may die from COVID-19, often 3-4 weeks after becoming infected. So it's logical that these things are always somewhat behind. Now, in order to get a better handle on what's happening in real time, one can calculate the reproduction number. That is basically how many people does each case infect. And and you know, the magic idea is that if we go below 0, below 1, each infected person infects on average less than one other person, then over time the infection will go away, and if it's higher than 1, then it will spread further. So that is something that will, you know, allows us to look into the near future. And things become even more complicated. It's not just the numbers of patients with infection and the other conditions, it's also the virus, which is not static, but it adapts and it changes. Most notably so, and something that South Africa can be proud of, if you know, in that context, that's the right word, was the end of in December 2020, when the new variant 501YV2 was discovered by us, by the NGS-SA consortium. It arose first and was first noticed in the Eastern Cape. From there, it spread along the coast, both northwards into KZN and west into the Garden Route and then into Cape Town. And these variants are a worry, and we always ask ourselves whether a particular variant is more easily transmitted, whether it causes more severe clinical disease, and if it poses an increased risk of reinfection, so meaning somebody who's had COVID before might become reinfected with this variant. In future, we also need to worry about these variant viruses infecting people who've been vaccinated because of what we call immune escape. So there are various features of these variants that are worrying. And if you look here at these are the sequences from the Western Cape. So it's us at Tygerberg Virology as partners in NGSSA and our colleagues at Grooterskloof and UCT who have generated these data. And you see that we started early with sequencing, low numbers, increased as the second wave came into being. And you see these here, the YV2 variant, it's the purple ones here. You see how it was first found in the Western Cape in mid-October 2020. Early November, it was already the majority of cases, and from the end of November, we've seen hardly any other variants at all. And that, of course, is a strong indication that this variant has some properties that make it superior, fitter than the other ones. It basically displaced the other variants. So, you know, and we know that the viral load is higher in people, and it's probably more transmissible. So now, how do we tackle this? How do we look into the future and monitor a possible resurgence? And we are actually now in a period where we are very eagerly looking every day for evidence of the beginning of a third wave. It looks like it. Unfortunately, all the indicators are showing, and there are different definitions of cases over time. One has to take into account certain factors like holidays, pauses in testing and so on and so forth. But, you know, it unfortunately looks like we are moving into a third wave. So what I have tried to show you in terms of an introduction to tonight's topic is that COVID-19 is not easy. By the time somebody comes forward for testing, they have been infected for several days and kind of could have passed on the infection already. Many cases go untested, even though they may have symptoms, particularly during these epidemic peaks when people are actually discouraged to come forward for testing because the health systems can't cope with that. And then, of course, there are many infections out there. Some people estimate maybe as many as 50% or even higher that show few or no symptoms, and where people may simply not be tested because they don't think they are ill. South Africa, and particularly Western Cape, I believe, with all the negative news and ideas that one has, I think we can be proud of having a very good epidemiological surveillance system. I think this is on par with the best in the world. And that's not only genomic surveillance, as NGS-SA is performing, but also the other arms of surveillance, counting cases and so on. I think this is really as good as it gets. However, none of the parameters that are assessed is perfect. And I think I've been able to show that there is undercounting and there are other factors that change over time. Still, taken together, they do offer good insights, but in terms of early warning, they can really only tell if there is an impending surge for the healthcare utilization. So that, you can say, well, we think in a week from now, we will have more patients needing hospital admission and needing oxygen, but we can't look further ahead.
Later for 5 minutes.
I'm now already done with my talk. So thank you very much, and I would like to hand over to Edward Archer for the second lecture. Thank you.
Thank you very much, Wolfgang. So Edward Archer is from the Department of Microbiology, and he's the coordinator of the Success Network. Edward, please go ahead.
Edward, you're muted.
I'm so sorry. Can everyone hear me now? Yes,
we can.
Okay, great. Yeah. No, so thank you for the introduction, and also the great overviews shared by Professor Preiser. What I'll be doing is to basically share the insights on where this concept of wastewater-based epidemiology came about and how it's being implemented in trying to tackle the cases or the issues that are being seen with this current pandemic on the spread of the virus, and also this asymptomatic issues that are being foreseen, as well as the new wave that will be coming in. So, as Professor Preiser has clearly shown, with the diagnostic testing coming into an infected person, where various biological or chemical stresses can be introduced into a person, diagnostics then basically sits in the forefront to look at these biomarkers of exposure. So these can either be proteins, chemicals, or then molecular markers such as DNA and RNAs that has been shared here in this previous presentation as well. So if we consider these biomarkers of exposure and where this concept of wastewater epidemiology came about, on the chemical side, we know there's consumption and also disposal of these chemicals by an individual. Various problems actually arise from chemical usage, where people over or abusing certain drugs won't actually be shown or actually be detected because they don't come up in police seizure reports or being admitted in rehabilitation centers, for example. And mostly this kind of analysis has been done on illicit drug analysis using wastewater to look at community-wide infections. So if we consider, for example, a infected person or a person that's being administered with a stressor, we can now also think of this one person as a whole community, that various cohort of households that's connected to sewer lines basically acts as one person excreting a sample that can be diagnosed. So this comes in different lines from communities and then being sent to wastewater treatment works where various of these biomarkers of exposure can then be detected and quantified or analyzed and looked at over a temporal and spatial scale. So, as I mentioned with the tracing of chemical substance abuse, how this actually came about with wastewater epidemiology, this then also allowed to look at these chemical markers and what is being excreted in the communities, and actually looking at the various levels as it proceeds throughout the week. Such as over here, presented here for cocaine, for example, MDMA, that shows a recreational drug use trend over a weekend period, and other drugs that are non-recreational. So it really shows the power of using a community-wide assessment of a stool sample, basically, as you could think, to see what's happening in the area. And this was also showing quite a lot of value in tracing, looking at the changes in substance usage, for example, where we've sampled, for example, in 2019, and then within the lockdown period of this current SARS-CoV-2 pandemic, and actually also saw a decrease in the recreational drug use trends. So actually showing a lot of value on how this approach can by using all these markers, can actually show us what is happening in the areas. And then I'm giving away my age over here, but we don't have to wait until September or last September or last summer to actually see what was happening in the community. This is a nice way of looking at the previous day or within a near real-time mode, what is going on in the communities. So with this concept of wastewater-based epidemiology being implemented for molecular markers as well, as previously mentioned, it's also been implemented a lot for other infectious disease. But then for the current SARS-CoV-2 surveillance, a huge cohort of collaborators actually came about at the start of the pandemic to work together and actually develop this cluster of epidemiology and analytical approaches to actually look at the value of this kind of methodology to look at community-wide infections. So similar to the usage of biomarkers of exposure then for chemistry, the molecular biomarkers can also be found on these viruses that are being shed by people. And this is an example of certain biomarkers of exposure that can be analyzed within a community. So if, for example, there's infection inside certain areas, the ribonucleic acid or genomic fingerprint of a viral infection, not the virus itself, but rather its fingerprint will be detected in a wastewater treatment plant, for example, and then community-wide trends can be looked at over a temporal and also spatial scale. So this then holds the value to move a bit upstream as well, not just from the community-wide or the total catchment area of what the wastewater treatment plants serve, but also then trying to look at defined communities or defined locations to actually narrow down and try and see what's happening in certain regions to prevent spread. So this is just a quick overview of the methodology on how the samples are actually being received by the South African Medical Research Council, and Rabia will give some results on this, and I'll just quickly explain the procedure on how we get to this stage. So at the forefront, sample collection is always an issue because there's a lot of logistics involved and a lot of issues to actually obtain the perfect quality and quantity of what we're trying to look at in the wastewater. So various modes of collection is being done, then transported and processed then at MRC for our case, with the studies that we're involved in, and quantification of the viral genome, and then reporting of the data to end users and stakeholders. One big objective here is to either decide whether we want qualitative to get a yes, no, presence, absence of the virus, or maybe move to a quantitative state where actual values and concentrations can then be obtained. And similar then to conventional wastewater epidemiology approaches, this quantification can then be back calculated or further normalized to receive in the end a per capita loading or per capita cases within the different catchment areas to compare between one another and see what is going on. So this then involves a lot of getting down and dirty procedures to actually go and try and capture the content that is being sent for analysis, and various modes of sampling is being implemented and also supported by various funds and collaborating bodies to actually make this happen. So on the left-hand side, you'll just see an example of how sampling is being done at various manholes at defined communities, and also over here collection at wastewater treatment works using composite or grab sampling devices. So Dr. Johnson will be presenting on some of the results that is also obtained from looking at defined communities. And one of our biggest proof of concept studies was now implemented at the Tygerberg Medical Campus from Stellenbosch University, and then selecting specific locations of manhole covers where sewage is being flown that comes from certain regions or residences. And more recently, there was also now the implementation of this approach on the Stellenbosch University main campus to get this proof of concept running as well, and which holds quite a strong value because we sit with a much younger generation inside this, where quite a significant percentage might be also presenting asymptomatic rather than going and being diagnosed for infection, and therefore we need to increase this kind of methodologies in order to prevent further spread of potential hotspot area or super spreading events. So with that introduction, I'll just end this presentation and then pass it over to Dr. Johnson for the analysis part.
Thank you very much, Edward. Rabia, over to you.
Thank you very much. And Edward, thank you very much for laying the foundation, as well as Prof Wolfgang Preiser. So this, I must start off by saying I've never ever seen the science community working together so efficiently and effectively as what I've seen in my time in science during this COVID-19 pandemic. I just want to give you a brief overview and show you how SAMRC, with all the different partners that Wolfgang Preiser as well as Edward introduced you to. So we know that the first COVID positive case in South Africa was in March 2020. That was also the same time that we've observed that reports came out that you can actually detect viral RNA in wastewater. During March as well, more reports came out to say that you can actually detect the viral RNA in wastewater a week prior to any new positive case data. And since then, the early warning system for the SARS-CoV-2 was established. In May, there is a group of SAMRC scientists as well as scientists from Edward's department and Stellenbosch University came together, and we started our first planning session to see can we use this as an early warning system to track and trace the current COVID-19 pandemic. In June 2020, we
Rabia, sorry to interrupt you. We cannot see any of your slides. I don't know if you're sharing or.
Oh, let me see what's happening here. Can you see it now?
Still not, no.
No, let me see. What's going on?
Okay.
Can you see my screen?
Okay. No, I can see it.
Okay, my apology. So in June 2020, we did our first proof of concept study, and that was also published. And in July 2020, we decided that was when Cape Town was at the epicenter of the first wave. We did a 6-week longitudinal study. In July 2020, we decided, wait, let's get all the other provinces involved, and we started training so that they can implement a similar early warning system within the different provinces. And in April 2021, SAMRC now said that we are actually an official SAMRC research program, of which Stellenbosch University is part of it. So basically, it would, like I said, lay the foundation, and like it would explain that this is a historically old methodology that has previously been used for the detection of various other viruses like your polio, measles, and hepatitis. And now we're using the same technology and methodology to track and trace the current COVID-19 pandemic. And like I explained, we know that both in symptomatic and asymptomatic patients, the viral can be shed. This ends up in our sewer system. This then goes into our wastewater treatment plants, and this is where we come in as the laboratory-based team, where we collect the sample like Edward showed you. The sample is then taken immediately to the laboratory, and we make use of a simple quantitative real-time PCR technique to detect and to quantify the viral load within that sample. So why is this important? This is important because if you look at this specific map, you'll see these are various wastewater treatment plants mapped out. Now, if you look at this specific diagram, this wastewater treatment plant is linked to various suburbs. So if we can detect the viral load within those different wastewater treatment plants, we can use this as an early warning system because number one, we know we can detect it early enough. And number two, we also know by looking at the different wastewater treatment plants, we can see where's this high viral load, and we can then use this to identify hotspots. And like I said, more importantly, is the fact that if you look at this graph here at the bottom, the red graph here and the black one, the red one showed, this is a study done by Pecker at Yale University, where they showed that the red is the viral RNA detected in wastewater. You can detect that 7 to 14 days prior to any detection of a new case. And Professor Preiser also alluded to that to show that the person is infected and will shed the virus even before the symptoms appear. And that's how wastewater-based epidemiology can be used to track and trace the current COVID-19 or new COVID-19 outbreaks. So that's why if you look at this graph here, so we've gone through the first wave, the second wave, and now we are awaiting the first wave. And the question on this forum now is, can we use this as an early warning system to predict any possible new resurgence or increase in COVID-19 cases, but using the wastewater? And can we thus use this to predict the first wave? So my answer to that is most probably yes, and I will show you data that confirm what I just said now. So what we did was, like I said, SAMRC has various sites. We've got sites in Western Cape, Eastern Cape, Gauteng, as well as Limpopo area. So what happens, the sample comes in, Edward and his team will bring in the sample. That sample will come in on a Monday, and on a Tuesday, on a Monday, that sample will be extracted, and on a Tuesday we will perform the PCR analysis. Now you must remember the PCR analysis is exactly the same PCR analysis that will be used for the COVID-19 case numbers. The only difference is we look at viral load as well. Thereafter, on a Wednesday, this is then sent to our team who will do the spatial mapping and also prepare the samples for our stakeholders, which is the different municipalities that we are reporting to, as well as the Western Cape government. And we know this also forms part of Keith Cloutey's weekly Digicon. And thereafter, this data is then put onto our dashboard that Professor Preiser was referring to. So I will give you some information of our 6-week study that we did during the first wave. And here we were looking at 24 wastewater treatment plants over a 6-week period. And this was done, like I said, during the first wave when Cape Town was at the epicenter. We started in July and we ended in August at the tail end of the first wave. So here you can look at the mapping of these different wastewater treatment plants with the legends. If you see red, it means the viral load was quite high, and this corresponded to the case numbers, with the green to being your lower viral load. So here you can see, if you look at the mapping of our cases of our viral RNA that was detected in the wastewater treatment plants, for the 24 wastewater treatment plants, you could clearly see during the first and the second week there was high viral load. As we moved towards the August, we could see more greens, which means the viral load was low, and this actually matched our case data. I'm just going to select one of these wastewater treatment plants. So what happened is, if you look at the red line is your case numbers, and the dotted line here, that is your viral RNA that was extracted from the wastewater with COVID, that was COVID positive. So here you can see there was a decrease in the viral load, and that followed by a decrease in case number. If you look here, from the 15th of July onwards, you could see there was an increase in the viral RNA within the wastewater. Similarly, we saw a similar increase in the case number. That was followed by a decrease in the viral load in the wastewater, and we saw a decrease in our weekly case number during that time. Something else that's also interesting using wastewater-based epidemiology as an early warning sign. I know the slide is busy. I want you to have a look at the red bars. These red bars are the level 1 restrictions, whereas the green one, the red ones is level 3, and the green ones is the level 1. And I also want you to look at the graph for the red, that was Oudekraal wastewater treatment plant, and the yellow will be your Millis Point, and that was our negative controls, because during your hard lockdown, Oudekraal and Millis Point wasn't open. So it was quite interesting with this data was over the 6-week period when we mapped it, we found that we couldn't see any SARS-CoV positive cases in these wastewater treatment plants. And then all of a sudden, when we moved to level 1 and everything started to open, if you look at this red bar here, which is your Oudekraal, all of a sudden we saw the spike. Similarly, when we look at Millis Point, here we could see there was also increase. Then we went back into level 3 lockdown, and all of a sudden it disappeared again, that we didn't see any positive viral load or viral RNA within our wastewater treatment plant. And I didn't map now our current data in here, but just shows that the wastewater epidemiology is actually working. We are able to track and trace and detect the viral RNA within the wastewater. So everybody will ask the question, so what is currently happening? Are we seeing, is the third wave here? So this is our data from 21st of December. This is data from our second wave. And this is hot off the press. This is this week's data for Western Cape only. Tonight I only presented Western Cape's data. And here you can clearly see that we are slowly but surely seeing an increase in the viral load within the wastewater, but there was no major spikes that we are currently observing. So we just change our legends a bit here. So all these dark blues is actually similar to the greens that you are seeing here. So we are actually seeing low viral load, but it's increased compared to previous weeks where we actually didn't detect viral RNA or SARS-CoV-2 viral RNA within these wastewaters, but now we are seeing it's creeping up on us, where we see low viral RNA in these wastewaters. For example, last week there was about, we are screening 24 wastewater treatment plants within the Western Cape area, and we were able to detect N equal to 8 wastewater treatment plants that was SARS-CoV-2 that was last week we found that there was N equal to 12 SARS-CoV wastewater treatment plants that was negative for SARS-CoV-2, and this week there was only 8 that was negative, which means more and more wastewater treatment plants are showing positivity for SARS-CoV-2 once again within the wastewater treatment plants. Edward was referring to the defined communities, and this is just a nice example of where we can show that within defined communities how we can track and trace again. So this is a map of Stellenbosch University Tygerberg campus. The DF that you're seeing here, this is the manhole at the right at the where the securities are sitting currently. And if you look at the DS3 here, this is actually a manhole that comes from the Huis Ubuntu Meerhof, from that line there. So if you look at the bottom, you can clearly see we started to track and trace SARS-CoV-2 viral RNA within this manhole starting the 20th of the 10th, and we couldn't see anything. And all of a sudden, we saw a spike here. And then you could see, okay, there must be some positive cases there. But unfortunately, at that point in time, we was looking at DS4, which is right at the end where everything will flow into. So what Edward did then, what he did was he actually went to DS3 because that specific line comes directly from Huis Ubuntu, as well as he looked at DS4, and this line would come from the specific clinical education building. And what was quite interesting was we found that we could see a positive signal coming from here, DS4 on the 3rd of the 11th, but not from the student residents if you look at the 3rd of the 11th. But what was quite interesting, what Edward also found was when he collected a sample during the December period where there was a graduation ceremony going on, we also saw that there was an increase in the viral load for your DS3, which is the line that actually comes from your Meerhof and Huis Ubuntu. So this is another way of how we can actually look at your defined communities and track and trace to see if we can pick up viral RNA within manholes. Then something else that was also quite interesting was this is ZANDVLIET is one of the wastewater treatment plants, and continuously throughout February, March, April, we found that we saw high viral load within the ZANDVLIET wastewater treatment plant. Once again, if you look at this map here, so what Edward then did was, we know that the wastewater treatment plant, say this is the ZANDVLIET wastewater treatment plant, and there are various lines that flows into the ZANDVLIET wastewater treatment plant, and we found that both the Blue Downs, Kalicha, and the Momabezi lines flows into that specific wastewater treatment plant. So what he did was he collected samples for us from Blue Downs, Kalicha, and Momabezi. So if you look at the ZANDVLIET, it's the green, and quite interesting when he collected from those specific plants, he found that high viral load that we were observing actually came from the Kalicha area and not so much from the Blue Downs or the Momabezi area. From here onwards, we also can use wastewater-based epidemiology using a simplistic PCR method to detect the current variants that's circulating. So this is just a quick map of how you can read the data. So everything on the y-axis, if you look here, that will be your mutants. And anything to the far right on your x-axis, that will be your wild type. And so what we found is we specifically just, so what we're doing now, so any positive COVID-19 viral load that we detect in the wastewater, we would then, the next step would then be to do a variant PCR on that to see what is currently circulating. And we found that for most of our wastewater treatment plants that was positive for SARS-CoV-2, we found that most of it is the 501 variant that's circulating there. For example, if you look at the Borchards Quarry wastewater treatment plant, we could see it's the 501 variant circulating there. And however, what was quite interesting from the ZANDVLIET area, we found that the ZANDVLIET came up positive, not for the 501, but for the 6970 deletion. And then we decided, wait, let's go look at the Kalicha, because Kalicha flows into ZANDVLIET. And voila, what did we see? We found that the deletion was present there and not the 501 variant. This is just to show that it's actually working. So from here, this is just my take-home message. And to summarize, I can just say from our wastewater data, that's supposed to give us a 1 to 2 weeks lead time to any increases. I can say that within the Western Cape, we can see that there are no major spikes, but definitely we're seeing that there are increase in the SARS-CoV-2 positivity within the various wastewater treatment plants, whereas previous weeks, our data, we didn't really detect high viral load, and we are currently seeing these increases. No major spikes, but we are seeing that something is currently happening. And then I would like to say thank you to all the funders and to even Stellenbosch University Science Café for giving all of us the platform to showcase what is currently ongoing. And I would also like to say thank you very much for the science community. I've never, like I said in the beginning, never ever seen us coming together like this to fight this specific pandemic, and it just show how quickly one can move forward if we all work together. Thank you very much.
Thank you very much, Rabia. That was a fascinating talk, and thanks also to Wolfgang and to Edward. I think in combination that is really a fascinating overview of a very important area with a gigantic impact on public health. We have about 15 minutes at our disposal to take questions and for discussion. There seems to be a little bit of a problem with the chat function. So if you do have questions, you're very welcome just to put up your hand, and I'll take the questions. Cornelia Maladba did manage to actually submit a question via the chat function, and I guess it's directed at Wolfgang. She says, thank you for the informative talk. Is there any form of tracking of illnesses caused post-COVID infection that is a result of COVID? If yes, is it considered to have a substantial burden on our health system as well?
Thank you very much. That's a great question. You may have noticed, and you're putting your finger into the weakness there, the numbers of recovered cases that many national accounts also include. This is not a real metric. What that means is people who've been diagnosed minus the people who are known to have died minus the people who are still in hospital, and then give it 2 weeks or so. So it's an indirect measure, and it includes people, even though it says recovered, it includes people who have what has become known as long COVID, which seems to be quite a significant negative consequence of COVID, affecting adults, even people without risk factors, even children who may or may not have had a significant COVID disease. So even people who have had mild disease have been described as suffering from long COVID. There are many unanswered questions as to what exactly it is and how it is diagnosed. It's very much a diagnosis of exclusion at this stage. I think in terms of therapy, there is a lot still to be discovered. Also, how permanent it will be and when it will be over. But I think this is one of the negative consequences of the pandemic that we are yet to grasp fully. So a great question, and it's something which is not being counted, not in these usual metrics. So it would require, and there are such studies, a different approach to do that. But it's none of the numbers that one finds would reveal that. Thanks.
Good, thank you very much, Wolfgang. The next question is from Dr. Blichnoot, and the question is, to what degree does viral load in wastewater correlate with patient numbers? I think any of the three of you are welcome to have a stab at that. To what degree does... Yeah, please go.
I will start with, and then maybe Edward, because that is what we are currently trying to work on. And there you need your population estimates as well as your flow rate, that we can use that to normalize again the current viral load that we detect with our PCR. We need that, and to normalize that before we can actually do a proper correlation. And that is what's currently taking time, because we are struggling to get hold of the case numbers. We're also struggling to get hold of the flow rate within the different wastewater treatment plants. So that impedes without doing a correlation as such as I showed you, what we found that for the smaller wastewater treatment plants, it's easy to do the correlation, but for the bigger ones, we are struggling, especially now during level 1, where people are actually moving around. And we sometimes, for the bigger wastewater treatment plants, we don't see a nice correlation, as I showed you with, like, for example, the example that I use, which is a smaller wastewater treatment plant. Edward, you want to add to that?
Yeah, thanks, Rabia. Yeah, it's clearly stated, as you mentioned, that this, the biggest issue with this kind of normalization is also that you don't know the amount of shedding that actually happens per person. So an infected person will have various levels of viral shedding of these fingerprint or biomarkers. So per capita normalization really requires a lot of geospatial mapping and actually understanding, and this is where the Success Network actually came in quite strong with our epidemiologist collaborators from UCT, for example, that has been using GIS-based databases to actually try and track and trace the movement of individuals within catchment areas and also try and capture what is the total population inside an area, because all of this kind of wastewater data is based on census data that's quite outdated since 2011. So estimating the amount of population that's within a catchment is really showing a lot of improvement with using chemical markers as well in wastewater, such as using ammonia or chemical oxygen demand, and all these different markers needs to be normalized to show the amount of shedding per individual. So this is quite a big headache that is still a universal problem on back calculating, getting correct population normalized estimates for each catchment. So this is where our modeling team also comes in.
Thank you, Edward. Wolfgang, you had your hand up. Do you want to add to that?
I think it's been mostly answered already, but just an sort of an experience. I'm learning as we go along, and I must say this is absolutely amazing. It is the first envelope virus which is not naturally, out there, transmitted the fecal-oral route that is detected as a marker in wastewater. As far as virology is concerned, it all started with these very hardy polioviruses that are by their very nature, that's their ecology, is that they are shed in the feces of the infected person and then through contaminated drinking water infect the next person. And they are very hardy. They can remain infectious for many weeks and months out there, even in sunlight and in the presence of all sorts of inhibitory substances. So it's absolutely amazing that this envelope virus is also can be retrieved from the wastewater. But there are interesting factors. Remember the wastewater treatment plant that receives the airport area here in Cape Town with the Monday peaks, and that is when the portable toilets, the portaloos from any informal areas are emptied and cleaned. So they are taken there, there's a yard, and they are cleaned and emptied, and then there's a spike, of course, and that means it's not being produced there, it comes from elsewhere. Lots of factors to be taken into account. Is absolutely fascinating. Of course, also dilution factors in terms of industry. Industry that's also using the wastewater system and dilutes the human feces. So it's highly interesting, and I think one has to be aware that people will continue shedding the virus for variable periods of time after the acute infection has cleared. We see that even in the throat swabs, where people may be positive by PCR with a sensitive method for several weeks sometimes after the infection has cleared, and the same would go for feces. So we have a lot to learn still. Thanks.
Thanks very much, Wolfgang. There's a question from Mr. S. Burgess. He asks, could one use this data to potentially decide on lockdowns for certain areas or districts? Go ahead.
So current, at this point in time, no, it's not being used, but that's the whole idea why we want to set this up for any future outbreaks, and we want, and the only problem is what we've just discussed previously. whether or not it match directly with the current case numbers because of people's movement. But I know it's currently being incorporated in the Cape Town's strategy for waste. In the current strategy with Keith Cloutier. But it's not there yet, but that is where we would really want to move towards. Prof Preiser, I don't know if you want to add to that.
If I may, yes, please. Yeah, I think it will be, and it is actually already incorporated as such in the Western Cape. It will be an additional marker. I don't think that wastewater data on their own would, justify locking down an area, but it is clearly an early warning marker which needs to be seen in conjunction with the other data. But I mean, remember when surges happen, like we saw this catastrophic second wave in December, which almost overwhelmed even the Western Cape's health system, which is the strongest in the country, which was very well prepared, and even we got to the limit of our oxygen supply. So even knowing a week in advance that in a certain area there is trouble waiting, that would allow one to take precautions. So I think it is valuable. Another experience that we've made is that, of course, the wastewater network is not aligned with the health district distribution. So to match the data, normally it's per district. The clinical data. There's a very good database in the Western Cape. But it does not necessarily overlap 100% with the wastewater network. So again, one needs to, it's quite intricate to interpret this. There are sort of areas that feed into a different wastewater stream. It's absolutely fascinating. I have to say that the European Union just issued, and I'm not sure what it's called, a directive or something to their member states that they must start introducing and conducting wastewater surveillance. And I know from my hometown, Frankfurt in Germany, that there's been debate for many months, and there's been a trial, and then it's been abandoned again. I think we are actually quite at the forefront of this, and it's something that poses a number of challenges, but it's ultimately I think it's fantastic to explore these modern ways of improving our epidemiological data. Thanks.
Thank you very much, Wolfgang. There's a question from Juliet Pulliam from SESIMA, who asks, does the presence of 501Y.V1 in wastewater suggest that there's widespread community transmission of this variant? Wolfgang, go ahead.
It's the excellent question. I don't know. I think we don't know about the relative the proportion of these different variants in wastewater. But what it does already when we saw this data, when Rabia found that she's picking up this deletion, which is not known from the South African variant, that we now homed into sequencing patient samples from that area affected. And I think that is one of the advantages. So, and we had not seen that. We are conducting molecular surveillance on an ongoing basis, but that of course it's a lot of work. It's expensive, so we can only sample a minority of positive specimens. And so one needs to know where to look. And we have all those vaccine breakthrough infections, reinfections. We have localized outbreaks, super spreading events that need to be sequenced. So, it's very valuable to get a warning that, okay, now this is an area from which you should actually sequence the next few samples, and that is happening, but we haven't got the data yet. So I cannot tell whether we've already now going backwards from the wastewater into patient samples, whether we've picked it up yet or not.
Thank you very much, Wolfgang. So there's a question, I think we've run out of time. There's a question from Penny van der Bank. Penny, could you perhaps just directly email that to Wolfgang Preiser? He'll be happy to read your email and respond to your question. I think at this point, what I want to say is we're at the end of this evening's Science Café. Time always flies when you're having fun. Thanks to all for attending. As we indicated in the beginning, this meeting is being recorded. We're going to stop that now. If you'd like to receive this recording, please indicate that in the chat, and please make sure that we have your correct contact details or email address. And if you're struggling with the chat function still, I'm sure an email to Guido would be welcome. In a face-to-face meeting, we would have invited you to stay on for a drink or snack and time to socialize. I assume you came prepared and have that ready wherever you are now. So a very good evening to you all. Please keep safe and see you at the next Science Café. Good night.